6.5 Coleoptera. General characters. Head. Head is strongly sclerotized, pro or hypognathous, mobile with reduced or absolute epicranial suture. In some beetles an elongated rostrum is formed due to prolongation of frons and vertex. Rostrum possesses mouthparts at the apex and antennae elsewhere in the forward position. Antennae. Antennae are usually 11 segmented and of variable type filiform, maniliform, clavate, serrate, lamellate etc. Compound eyes. Distinct, except in some subterranean and caverniculous forms, where the eyes are totally lacking. The eyes are of three types: acone, eucone and pseudocone. Ocelli, usually absent while only a median ocellus is present in dermestity. Mouthparts. Biting chewing type. Labrum. Concealed beneath or fused with clypeus. Mandibles. Variably developed. In carnivorous beetles they are acute with fine cutting edge and canaliculated for fluid feeding, while in phytophagous forms they are short, broad, bluntly toothed. They have serrate edge and are longer even more than the whole body. Maxillae. Fully developed, bilobed while only a single lobe or mala. Maxillary palps are four-segmented, lacinia are blade-like. Labium. Variably lobulated. Mentum fused with gula and glossa paraglossa are fused to form ligula. Labial palps are three-segmented. 110. Thorax. Prothorax, freely movable with large, free, visible, single undivided pronotum. Notopleural sutures absent in Polyphaga, pleurosternal sutures are distinct. Meso and metathorax reduced, fused anterior terga are divided into pre-post scutum, besides the middle scutellum. Legs. Modified widely walking, jumping, running, fossorial, natatorial. In aquatic forms medanned hindlegs or only hindlegs are modified for swimming. Wings. Up forewings, two pairs, unequal forewings modified into elytra. Both remain free, often open and allow the hind wings to perform flight. The elytra may be united and immovable in Carabidae, Curculionidae where the hindwings are absent. B. Hindwings. Larger than elytra, membranous, folded. Abdomen. Sessile, first segment membranous or atrophied. 8 tergites are commonly visible. 5 to 7 sternites are visible. Development. Eggs are laid in clusters or enclosed in cocoons, hydrophilidae, or in Uthica cassidinae, or in the tissues of plants, Curculionidae. Larva. With strongly sclerotized head, biting type of mouthparts, lacking abdominal legs. Thoracic legs well developed. There are two types of larvae. Campodiform in Adephaga, Eruciform in Polyphaga. General characters of family Hydrophilidae. This family includes water scavenger beetles. 1. They are aquatic or terrestrial, inhabiting decomposing vegetable matter. 2. Small or large, black or dull-colored oval, flattened and predaceous beetles including larvae. 3. Elongate maxillary palps performing functions of antennae. Antennae are short 6 to 9 segmented with hairy terminal clubs and function as respiratory organs. Tarsi of forelegs modified into clinging organs to hold female. Mid and hindlegs are fringed with setae. 4. Elytra encloses dorsal air reservoir and ventral hairy tracts which hold air film. E.g., Hydrus indicus, Hydrus opacus. 111. General characters of family Meloidae. This family includes blister oil beetles. 1. Body is medium size and elytra are brightly colored as metallic blue, green, black, brown, transverse colored stripes are common. 2. Head is hypognathous, strongly deflexed. Neck narrow but distinct between head and thorax. 3. Legs are long and with secrete tarsal claws. Tarsi are 5 segmented. 4. Wings are well developed, elytra cover the body incompletely and both are free. 5. Adults on disturbance, emit a defensive oily fluid containing canthardine chemical compound, from the apices of femur. 6. Female lay eggs in a hole under the ground. 7. Larvae develop in eggs of other insects. E.g., Malabris, Epicata, Lida, Mello. General characters of family Coccinellidae. This family includes lady bird beetles. 1. Adults are small, oval or spherical convex, brightly colored with red, black, yellow, brown spots. 2. Mostly carnivorous, feeding on aphids, coccids etc. Some are herbivorous. 3. Head is concealed in pronotum. 4. Tarsi are 4 segmented. 5. On disturbance, adults discharge a bitter amber-colored fluid having defensive function from tibio-femoral articulation containing dermal gland, e.g., Epilochna, Menachilus sex maculatus. General characters of family Curculionidae. This family includes weevils. 1. Head is modified into a long rostrum. 2. Antennae are often clubbed, geniculate or clavate. 3. Gular sulci are mostly confluent. 4. Mouth parts at the tip of rostrum. 5. Labrum is wanting, palps are reduced. 6. Body is clothed with scales. 112. 7. Mandibles, lacking or reduced. 8. Larvae are opodous and phytophagous. 9. Rostrum is used by female as a drilling instrument to bore hole for placing the eggs. E.g., Rhynchaenus, Pemphiurus affini. General characters of family Scarabaeidae. This family includes dung beetles. 1. Small or large, oval or spherical or convex body. 2. Head and prothorax with horns. 3. Antennae are 8 to 10 segmented. 4. Hind tibia with spur. 5. Mandibles are membranous. 6. They feed upon dung and roll the dung ball to a suitable place. The egg is enclosed in a small ball of dung in underground chamber and a nest is guarded by a female. E.g., Helicorpus, Cartharsisu, Copris. General characters of Chrysomelidae. This family includes potato, cucumber, flea beetles. 1. The beetles are of moderate size, oval, convex, black, blue, green, brown, yellow, metallic brightly colored beetles with beautiful spots or stripes. 2. Antennae are widely separated. 3. Dorsal surface of body is greatly plain and shining with metallic coloration. 4. Eyes are small. 5. Prothorax is laterally margined and narrow. 6. Elytra cover entire body. Phorecoxy is globose. 7. Legs are well adapted for running and jumping, e.g., leptinotarsa, longotarsus. General characters of family Cerambicidae. 113. This family includes long-horned wood-boring beetles. 1. Some species of adults attain largest body size among insects, e.g., Teotnus gigantus. 2. Body is long and cylindrical and with attractive coloration. 3. Antennae are very long, at least 2/3 of body length. 4. Tibia is with 2 spurs. Tarsi is 5-segmented. 5. Stridulatory organs are on hind margin of prothorax, scutellar base or hind femora, elytral edge. 6. Grubs are opodous, elongate, cylindrical, whitish and bore into woods, stems, roots of mostly forest plants and some domestic ones, e.g. Anaplophora verstigi, Stenias gricitor. 6.6 Summary. Hemiptera means, half-wing, is an order of insects commonly called true bugs. Majority of them are plant sap feeders, but some are predators and ectoparasites. There are several pests causing damage to various economics crops. Some act as vectors for a variety of pathogens causing diseases in plants and animals. Order Hemiptera is classified into 2 suborders, Heteroptera and Homoptera. In Heteroptera, head mostly hypognathous, mouthparts piercing and sucking type wings. The forewings are modified into the hemelytra. Hindwings are membranous, legs well developed, hindlobes longer, tarsi 2 to 3 segmented. Legs are modified into saltatorial, fossorial, raptorial or natatorial legs. Abdomen, it is 11 segmented. Sound producing organs among Hemiptera. Cicadidae are well known for sound production. In Homoptera, head is deflexed. In Homoptera, antennae mostly 4 or 5 segmented, mouthparts piercing and sucking type wings. The forewings are uniformly but slightly hard, legs well developed. Abdomen, it is 11 segmented. Coleoptera is the largest order in not only the arthropoda but in the entire animal kingdom, consisting about 330,000 species. They are adapted to various modes of life, viz., terrestrial, aquatic, aerial, fossorial, subterranean, carnivorous, etc., and are mostly concealed in habit. The order represents a heterogeneous assemblance of very minute size. In Coleopterans, head is strongly sclerotized. Pro or hypognathous antennae are usually 11 segmented, mouthparts 114. Biting, chewing type mandibles variably developed. In carnivorous beetles, they are acute with fine cutting edge and canaliculated for fluid feeding, while in phytophagous forms they are short, maxillae fully developed wings, forewings, 2 pairs, unequal forewings modified into elytra. Development, eggs are laid in clusters or enclosed in cocoons, hydrophilidae, or in Enuthecae cassidinae, or in the tissues of plants, Curculionidae. Larvae have strongly sclerotized head, biting type of mouthparts, lacking abdominal legs. Thoracic legs well developed. There are 2 types of larvae. Campodiform in Edaphaga, Eruciform in Polyphaga. 6.7 Terminal question. 7.2 Introduction. These are endopterygote insects having soft body covered with pigmented scales, with 2 pairs of membranous wings and suctorial proboscis, with a ruciform larva bearing 8 pairs of legs and with a decticus object pupae. It is one of the largest orders consisting of more than 105,000 species. The moths are nocturnal while the butterflies are diurnal in habit. They are beautiful insects representing species-specific color pattern of the wings. The adults feed upon nectar and juices of flowers and fruits, while larvae exclusively feed upon a variety of plants and are destroying foliage, shoots of trees, crops, vegetables, fruits, food grains, flour, clothing, etc. and behave as the external feeders or internal borers. The lepidopteran larvae are, therefore, pests of various economic crops and timber. The adults are, on the contrary, harmless except some species of fruit-sucking moths, such as the citrus fruit-sucking moth. The order Hymenoptera consists more than 100,000 species, and large number of them are still undiscovered due to severe parasitic mode of life. They may be free-living, phytophagous, predatory, entomophagous, parasitic, or social insects. Trophallaxis, or mutual exchange of food between adults and larvae, is common among ants and wasps. They have very diversified behavior, habits, nest building, caste system, chemical communication system, etc. Honeybees are the best natural pollinators and honey and wax producers. Chalcid, braconid, and ichneumony flies are well-known parasites and are widely used in the pest control management throughout the world. This method of pest control is referred as to the biological control. In various families 117 of Hymenoptera, particularly in the honeybees, some wasps and ants, the males are reproduced through parthenogenesis. Diptera is one of the largest orders of insects consisting more than 85,000 species. They are highly evolved in their biology. They feed upon nectar, decaying organic matter, microorganisms, on the tissues and blood of various animals. Some species, one or both sexes, spread or transmit various pathogens causing diseases in man and several vertebrates, farm animals, and thus acquire medical and veterinary importance. 7.3 Lepidoptera. General characters. 1. Head. Head is small, spherical and hypognathous, with a large frontoclypeal region. 2. Antennae. Antennae are long but of variable length, often clavate, pectinate. Plumose type in the males of Saturniidae. 3. Eyes. Eyes are compound, large, globular, lateral, concealed under scales, situated closely ventral to eyes. 4. Mouthparts. Mandibles are wanting. Maxillae both galea become elongated greatly, inner face become concave and are held together by hooks and spines so that a coiled suctorial proboscis is formed. 5. Thorax. Thoracic segments are fused but mesothorax is large and distinct. 6. Wings. Wings are two pairs, large, covered with scales on wing membrane. Microtrichia are common. Metathorax bears tympanal organs. Vestigial wings are even absent in the females of Psychididae. Wings are folded vertically upward, butterflies, or eld roof-like over dorsum, moths, at rest. Forewings are always larger than hindwings. Marginal lobes are common. Coupling of both wings are common. Longitudinal veins are numerous. 7. Forelegs. Forelegs are reduced. Coxae is large, long and immovable. Tarsi 5 segmented with a pair of claws. Legs are degenerated in the females of bagworms, Cyphalidae. 8. Abdomen. Abdomen is slender or tubular, 10 segmented, 9th and 10th segments in female form telescopic ovipositor. Tympanal organs are present at the base of abdomen. 118. 9. Male genitalia. Median sac is on the 9th segment with a pair of claspers, a median processor uncus, with edagus below gnathos and with a juxta or penis funnel. 10. Female genitalia. 9th and 10th segments form telescopic ovipositor. General characters of family Noctuidae. This family includes armyworms and cutworms, borers. 1. Wings with aerial, cryptic and dull-colored. SC free at base. Tympanum is present on metapomeron. 2. Proboscis is present, hind tarsi is slender, tibial spurs is long, frenulum is present. 3. Larvae are with all prologues bearing crochets except in Catocalinae, Plusinae and Hypeninae in which first two pairs are wanting and they become semiloopers. 4. Some moths are adapted for the habit of fruit sucking. 5. E.g., Acacia genata, Spodoptera exigua, Mythima separate. General characters of family Bombycidae. This family includes mulberry silk moths. 1. Male and female with pectinate antennae. 2. Moths are robust and large. 3. Larvae are large, wrinkled, white silkworms. 4. Body is cylindrical and feed on mulberry plants. 5. Larva with a short anal horn, mediodorsal on the 8th abdominal segment. 6. Pupa is formed under silken cocoon. 7. Wings, are without frenulum. Proboscis is vestigial or absent. E.g., Bombyx mori, Upterote, Nisiga. General characters of family Sphingidae. This family includes hawk moths. 1. These are large, robust, flying moths. 119. 2. Antennae are hooked apically. 3. Proboscis is extremely long. 4. Forewings are larger than hindwings. 5. Larvae have body segments, with longitudinal and oblique stripes. Larvae also have a horn on segment 8. 6. Pupation occurs inside cocoon, e.g., Acherontia, Deilephila. General characters of family Nymphalidae. This family includes emperor butterflies. 1. Butterflies are brightly colored with clubbed antennae. 2. Forelegs are reduced and vestigial, folded on the thorax. Tibia is short, covered with long hairs. Foreleg tarsi are unjointed in male but 4 to 5 segmented in female. 3. Larvae are covered with spines and a pair of anal processes directed backwardly. 4. Pupa is naked, e.g., Vanessa cardui, Nymphalis. General characters of family Pieridae. This family includes white, orange or yellow butterflies. 1. Forelegs are normal with bifid claws. Foretibia are without pads. 2. Antennae are clubbed. Forewing cu vein with 3 branches. 3. Larvae are segmented divided into annulates with setae. 4. Prologues with B or triardinal crochets. 5. Pupa is naked, attached to host by a silken thread from the anal end with a single ventral spine. E.g., Pieris brassicae, P. napi. General characters of family Populionidae. This family includes swallow-tail butterflies. 1. They are iridescent black, blue or green, spotted, yellow, orange-colored. 2. Antennae are clubbed. 120. 3. Large butterflies with tail-like prolongation of hindwings. Claws never bifid. 4. Tibia are with pads. 4. Larvae are without setae. 5. Pupa are with 2 lateral cephalic processes. Naked and suspended by a thread from anal end. E.g., Papilio demolius, P. polites. General characters of family Saturniidae. This family includes czar silk moths. 1. They are very large, colored moths, tropical. 2. Wings with transparent fenestrae or eye spots. 3. Proboscis is aborted, vestigial or absent. 4. Antennae are pectinate in males and females, long in males. 5. Frenulum is absent. 6. Hind wings with tail-like processes. 7. Larvae are large, smooth with scoli, with spiny tubercules. 8. Pupation occurs in silken cocoon. 7.4 Hymenoptera. General characters. Head. Head is free, mobile, hypognathous. Antennae, geniculate, clavate, filiform or pectinate. Segments 4 ants to 70 Ichneumonidae, sexual dimorphism 13 segments in males and 12 in females in Apoidea and Vespoidea. Eyes, well developed except in some ants where they are atrophied. Ocelli, 3 but sometimes absent. Mouthparts, biting chewing in Symphyta, lapping sucking in bees. Mandibles are well developed but do not serve trophic function and are modified for molding wax. Cutting leaves are 121. Tearing plant fibers. They carry out nest building in Apocrita. They are prehensile or feeding dentate organs in sawflies. Labial palps, 4 segmented. Glossa modified into proboscis bearing a spoon-like lobe, flabellum. Paraglossia vestigial or absent. Galea in sawflies modified into large blades. Thorax, mesothorax, largest. First abdominal segment tergite is fused with meta post notum forming propodium. Mesonotum is divided by a transfer suture into anterior scutum and posterior scutellum. Wings, 2 pairs usually present, sometimes greatly reduced, vestigial or absent. Forewings large while hindwings small. During flight, forewings are interlocked with hindwings by frenal hooks. A row of hooks are hamuli along the costal margin of the hind pair. These hooks catch on fold along posterior margin. Venation is greatly reduced in Apocrita while generalized venation in Symphyta. Total absence of venation. Platygasterity. Apteris, workers of ants. Apteris males of Aconity. Legs. Trochanter along with Trochantelis present. Apocrita forelegs with spur or calcar at apex of the tibia opposite to cavity of basitarsus. Basitarsus cavity with fine comb-like teeth which remove pollen from antennae and ladder are inserted and thus act as preening organ. Fossorial legs. Sphacoidea use for digging. Bees with pollen-carrying hindlegs, tibia with a large pollen brush or scope and margin with long hair forming pollen basket or carbicula while basitarsus forms preening organ, tarsi 5-segmented. Protarsi with aurolium and a pair of claws. Abdomen. First abdominal segment is fused with the metathorax forming propodium. Rest of the abdomen forms gaster of metasoma. First segment or gaster is broad and unmodified in Symphyta while modified into petiole in Apocrita. General characters of family Ichneumonidae. This family includes ichneumon flies. 1. Largest family, body is large, slender, black, yellow or brown insects. 2. Antennae are filiform, never geniculate but often long having 16 or more segments. 3. Hind femur is with a trochantelis. 122. 4. Forewings is with a pterostigma at distal end of costa. Costal cell is usually narrow. 5. Venetation is complete. Abdomen is with pygostyles. 6. Ovipositor is long, extruded permanently. 7. Pupation is inside cocoon. 8. Parasitism is exclusive on larvae of Coleoptera, Lepidoptera, Diptera, Hymenoptera, aphids. E.g., Amoromorpha. General characters of family Chalcididae. This family includes chalcid wasps. 1. It is one of the largest family of the order. 2. Minute or small metallic colored, primary or secondary parasites of eggs, larvae and pupae of Lepidoptera, Hemiptera, and Diptera. 3. Labrum is concealed. Mandibles are a tout with 34 teeth. 4. Antennae are geniculate, 2 to 13 segments, clavate. 5. Pronotum is not extending towards tegulae. Propodium is wanting from mesothorax. 6. Wings are not folded longitudinally. 4. Wingshai line are spotted, with marginal fringe, venation greatly reduced. 7. Ovipositor is not curved over the dorsum, short and straight. E.g. Myriella indica, Stomatoceras. General characters of family Vespidae. This family includes social wasps. 1. Red or yellow body with black markings. 2. Antennae is with 12 segments in females longitudinally, with a long, first discoidal cell, veins do not reach apical margin. Submarginal cells are 3. 3. Hindwings are without anal lobes. 4. Petiole is distinct long, slender. Pronotum extends back, lateral to tegula. 5. Mandibles are strongly dentate. Claws are simple. 6. Abdomen is sessile, conical, with well-developed terminal ovipositor or sting. General characters of family Braconidae. This family includes braconid flies. 1. Small, stout insects. 123. 2. Hindwings with a cross vein rem but differs in position from that in Ichneumonidae. Forewings without 2 M-Q cross vein, but, with M plus R's sector cross vein so there is only a single recurrent vein. 3. Larva is with 13 segments, with caudal appendage and endoparasites. Proctodeme in larva with swollen anal accessory respiratory organ. 4. Pupation is within cocoon inside or outside the body of host. E.g., Apantels, Microplitus, Stenobrachen. General characters of family Apidae. This family includes honeybees. 1. Small, black or brown, densely hairy, golden brown colored. 2. Hind tibia is without spurs. Clypeus is elongate, tongue is elongate. 3. Forewings is with 3 submarginal and 1 radial cells. 4 and mid-tibia are with apical spines. Hind tibia and metatarsus are flattened and fringed. 4. With pollen-collecting apparatus, best pollinators. 5. Sting is with terminal barbs. E.g., a dorsata, Apis indica. General characters of family Formicidae. This family includes black ants. 1. All are social and with worker caste. 2. Male and fertile female are primary winged castes, workers wingless, queen secondarily wingless after mating. 3. Antennae are geniculate, 4 to 13 segmented. 4. Labrum is vestigial. 5. Mandibles are excessive and variably modified. 6. Eyes and ocelli are developed in male, vestigial in male. 7. Wings are with 1 to 2 cubital and 1 discal cell. 8. Queen once fertilized, lays eggs and they often replace queens. 7.5 Diptera. General characters. Head, prognathous, freely movable, and relatively larger than the body. 124. Eyes, large, occupying most of the dorsum of head capsule. Both eyes may be fused polyptic, mostly in the males, or separated dicoptic mostly in the females. In some species eyes are larger in the males than females. Ocelli, mostly 3, arranged in a triangle. Antennae, Nematocera filiform in females, plumose in males, Brachycera styla type, Cycloropha aristate type. Mouthparts, sponging in non-blood-sucking Cycloropha or piercing and sucking type in blood-sucking forms. Females of Nematocera and Brachycera possess well-developed blood-sucking mouthparts while both males and females of some Cycloropha are well-equipped for blood-sucking. Labium universally forms the proboscis. Mandibles are absent or greatly reduced in non-sucking Cycloropha. In the blood-sucking Cycloropha, proboscis alone functions as piercing organ and the labella bear prostomial sharp teeth for cutting the tissues and skin of the host but lacks pseudotrachea. Thorax. Mesothorax is greatly developed, pro and metathorax reduced, with dense bristles showing characteristic arrangement. Wings. Single pair of forewings only, hind wings modified into club-shaped halters. Wings are modified into club-shaped halters. Wings are membranous, hyaline or colored, covered with setae or scales, venation reduced, 2A, 3A is vestigial and atrophy of cou is common. Posterior margin of wings at base possesses lobes, the alula and squame in many species. Legs. Slender or stout, weakly developed, mostly fit for clinging, tarsi 5-segmented, claws, paired, with empodium and pulvuli. Pulvuli are reduced or wanting in Nematocera or replaced by a single aurolium. Abdomen. Distinctly segmented but some segments or sternites are reduced. Female genitalia. Segments 6 or 7 to 10 often modified into telescopic ovipositor. Male genitalia. In male, 9 and 10 segments form a curved twisted hypopygium supplemented by a pair of claspers, edagus and with or without some accessory structures. General characters of family Tipulidae. This family includes crane flies. 1. They are large-sized nematoceran flies. 2. Antennae are multi-segmented, pectinate or serrate. 3. Head is anteriorly modified into a beak or elongate proboscis. 125. 4. Ocelli are absent. 5. Te mesothorax bears V-shaped suture on notum. Legs are very long. 6. Wings are often spotted. 7. Larvae have hemicephalous head, body is elongate, cylindrical, with or without pseudopods and trunk 11 or 12 segmented. 8. Pupa is elongated with slender or plate-like thoracic respiratory horns, e.g. Conocea irorata. General characters of family Chironomidae. This family includes midges or gnats. 1. Antennae or plumose in males and pelos in female. 2. Antennae are 15 segmented. 3. Wings are not covered with scales, anterior wing veins are thickened. 4. Mouth parts are poorly developed. 5. Forelegs are elongate, slender. Ocelli are absent. 6. Head is small, concealed in thorax. 7. Larvae are aquatic, red due to stored hemoglobin, with blood gills and 2 pairs of anal gills. E.g. Chironomus vicarius, C. cubicularum. General characters of family Culicidae. This family includes mosquitoes. 1. Body slender, long, without ocelli. Palpi is stiff, with elongate piercing proboscis consisting stylets. 2. Wings are fringed, scales along veins and posterior margin. 3. Legs long covered with scales. 4. Larvae are aquatic with respiratory siphon, metaneustic, with feeding brushes. 5. Anal segment with 4 tracheal gills. 6. Pupa aquatic and active. 2. The members are vectors of diseases like malaria, yellow fever, dengue etc. E.g. Anopheles. General characters of family Mussidae. This family includes true flies, houseflies, stable flies, buffalo flies and cattle botflies. 1. Small or medium-sized flies. 126. 3. Eyes are large, holoptic in males, rarely. 4. Mouth parts spawning in houseflies, sucking in others. 5. Antennae are with basal arista of plumose type. E.g. Musca domestica, M. nebula. General characters of family Tabanidae. This family includes horseflies. 1. Body is without bristles, flattened, stout, metallic brown, tawny or gray. 2. Antennae are without style. 3. Eyes are large, holoptic in males, dicoptic in females. 4. Females are bloodsuckers, males are nectar feeders. 6. Larvae are 12 segmented, small, with retractile head, with antennae and mouth hooks. 7. They are active bloodsuckers of cattle, horses and man. E.g. Tabanus striatus. General characters of family Drosophilidae. This family includes pomace flies, vinegar flies or fruit flies. 1. Small flies occurring about flowing sap, decaying fruits, wine, vat, vinegar fermentation products. 2. Eyes are red, antennae with second segment cleft, arista dorsally, pectinate. 3. Larvae are predatory, on fruits. 4. Body of larvae are 11 segmented and each segment is surrounded by a girdle of minute hook-like spines. E.g. Drosophila melanogaster, D. montium. General characters of family Bombylidae. This family includes bee flies. 1. Body is covered with pubescent hairs. 2. Proboscis is extremely long and piercing. 3. Legs are long and slender. Antennal style minute or vestigial. 4. Males with holoptic eyes. E.g. Bombolus major, Halictus. 7.6 Summary. The order Lepidoptera is one of the largest orders consisting of more than 105,000 species. The moths are nocturnal while the butterflies are diurnal in habit. They are beautiful insects. 127. Representing species-specific color pattern of the wings. The adults feed upon nectar and juices of flowers, and fruits while larvae exclusively feed upon a variety of plants and are destroying foliage, shoots of trees, crops, vegetables, fruits, food grains, flour, clothing etc. And behave as the external feeders or internal borers. Head is small, spherical and hypognathous, antennae are long but of variable length, often clavate, pectinate. Plumose type in the males of Saturniidae. Eyes are compound, large, globular thoracic segments are fused but mesothorax is large and distinct. Wings are 2 pairs, large, covered with scales on wing membrane. Wings are folded vertically upward, forelegs are reduced, coxae is large, long and immovable. Abdomen is slender or tubular, male genitalia median saccus on the 9th segment with a pair of claspers, a median process or uncus, with edagus below gnathos and with a juxta or penis funnel. Female genitalia 9th and 10th segments form telescopic ovipositor. In Hymenoptera head is free, mobile, hypognathous. Antennae geniculate, clavate, filiform or pectinate. Eyes well developed except in some ants where they are atrophied. Achilli 3 but sometimes absent. Mouthparts spitting chewing in Symphyta, lapping sucking in bees. Thorax mesothorax, largest. First abdominal segment tergite is fused with meta post notum forming propodium. Wings 2 pairs usually present, sometimes greatly reduced, vestigial or absent. Forewings large while hindwings small. During flight, forewings are interlocked with hindwings by frenal hooks. A row of hooks are hamuli along the costal margin of the hind pair. Legs trochanter along with trochanterus present. Bees with pollen-carrying hindlegs. Abdomen first abdominal segment is fused with the metathorax forming propodium. Diptera is one of the largest orders of insects consisting more than 85,000 species. They are highly evolved in their biology. They feed upon nectar, decaying organic matter, microorganisms, on the tissues and blood of various animals. Some species, one or both sexes, spread or transmit various pathogens causing diseases in man and several vertebrates, farm animals, and thus acquire medical and veterinary importance. Head prognathes, eyes large, occupying most of the dorsum of head capsule. Both eyes may be fused polyptic, mostly in the males, or separated dicoptic, mostly in the females. Antennae nematocera filiform in females, plumose in males, brachycera styla type, Cycloropha aristae type. Mouthparts sponging in non-blood sucking Cycloropha or piercing and sucking type in blood sucking forms. Thorax mesothorax is greatly developed, pro and metathorax reduced, with dense bristles showing characteristic 128. Arrangement. Wings single pair of forewings only, hind wings modified into club-shaped halters. Wings are membranous, hyaline or colored, covered with setae or scales, legs slender or stout, abdomen distinctly segmented but some segments are sternites or reduced. Female genitalia segments 6 or 7 to 10 often modified into telescopic ovipositor. Male genitalia in male, 9 and 10 segments form a curved twisted hypopygium supplemented by a pair of claspers, edagus and with or without some accessory structures. 7.7 Terminal questions and answers. Q1 Write some general characters of family Apidae. Q2 Write some characters of these families Vespidae and Chalcididae and how they are differ from each other. Q3 Write about the family of wasp which is known for their social behavior. Q4 Write some characters of family Tipulidae. Q5 Write some morphological differences between the family Chironomidae and Culicidae. 129. Unit 8, Insect Pests, Agro-Horticultural. Contents. 8.1 Objectives. 8.2 Introduction. 8.3 Origin of insect pests. 8.4 Factors affecting the abundance of insect pests. 8.5 Types of insect pest. 8.5 Types of insect pest. 8.6 Pests of stored grains, Cetophilus, Trogoderma, Rhizopertha, Tribolium, Bruchus. 8.2 Introduction. The term pest is derived from French word, peste, and Latin term, pestis, meaning plague or contagious disease. In other words, a pest is defined as any organism that causes annoyance or injury to human beings, human possessions, or human interests. The injury may be physical, bites and stings, medical, causing illness or disease, or economic, monetary loss to goods or property. Injury may arise directly from the pest itself, or may develop indirectly as a result of the actions or behavior of the pest. Being the most diverse and widely distributed animal groups on Earth, different insect species act as the pest to human beings in most if not all the above-mentioned categories. Specifically, several herbivorous insect species feed on one or many crop plant species and cause serious economic damage to important crop plant species and are considered as the major pest for agricultural and horticultural crop commodities. Insects populations increase above a certain level and cause economic losses to crop, fruit, product yields and stored products. In order to identify a pest species, an insect pest's population density is crucial. To reiterate, population density is a key idea that underpins knowledge of pests and their dynamics in agriculture. Even while there may be several different insect species present in a location at once, not all of them qualify as pests. Only those whose populations exceed a particular threshold and causes economic harm are referred to as pests. Such population densities of insect pests, which can cause economic damage to the crop, have been categorized into different levels. Concept of injury levels. 1. General equilibrium position, GEP. It is the average population density of any insect pest throughout a given time period and is unaffected by the pest control methods used on the crop. 131. Field. Population density at GEP does not significantly harm the crop economically for the majority of insect pest species. However, due to any biotic or abiotic reason, the population density at this level might vary and reach up to EIL. 2. Economic threshold level, ETL. It is an index of decision-making regarding pest management. Stern et al. 1951 defined the ETL as the population density at which control action shall be determined, initiated, to prevent an increasing pest population injury from reaching economic injury level. The ETL is essentially a time to take initial action, therefore even though it is measured in bug density, numbers are merely an index of that time. ETL is merely an action threshold since the level of harm produced by one insect, like that caused by other insects, can likewise cause significant crop damage. 3. Economic injury level, EIL. It is the lowest insect population in field which causes economic damage to the crop. According to Stern et al. 1951, EIL is the amount of injury which will justify the cost of artificial control measure. Preventive control methods are management strategies implemented at ETL stop the pest population before it reaches the point when crop damage is economically detrimental. Economic injury can be simply described in terms of the cost of pest treatment and the cost of yield loss due to pest infestation. Economic damage injury results when the cost of pest treatment is less than the cost of yield reduction. 8.3 Origin of insect pests. Insects have been believed to be emerged roughly 480 million years ago, during the Ordovician period, around the same time as the earliest terrestrial plants. The coexistence of the two types also gave rise to herbivora, one of the most significant co-evolutionary phenomena. After that, the appearance and subsequent fast success of blooming angiosperm species throughout the Cretaceous period led to the creation of insect societies, ranging from ant, termite, and nectar-feeding bee communities. Insects that consumed plants acquired a variety of mouthparts during the course of the following many hundreds of years, from biting and chewing to piercing and sucking varieties. On the other hand, humans first appear on Earth nearly 6 million years ago during late Miocene epoch of the Cenozoic Tertiary period. As humans during early phases of evolution did not grow any crops and did not know the concept of homes and had nothing to very few possessions, we 132 can imagine that their pest problems would have been limited to the insects, such as lice, fleas, flies, and mosquitoes, causing different fatal diseases. Several examples of such insect-transmitted diseases, plague, have been reported in prehistoric and historic period of most if not all the human civilizations. It was only after the development of agriculture, approximately 10,000 years ago, the establishment of permanent settlements, and the introduction of a lifestyle that required the storage of greater or lesser quantities of food and other items that a concerted effort to control a large variety of organisms became necessary. Early pest control practices were often based on mysticism or superstition, but some of the evidence from the Chinese ancient texts reports for different pest management techniques of historic periods, some of which are still in use. The Sumerians were utilizing sulfur compounds to keep insects and mites under control well before 2500 BC. Plant-derived insecticides, similar to modern botanicals, had also been produced for seed treatment and fumigation purposes by 1200 BC in China, thousands of kilometers to the east. For the prevention and management of pests in buildings and on stored goods, the Chinese also utilized chalk and wood ash. Similar methods of pest control can be found in Greek and Roman history. Homer recognized the benefits of burning for locust control about 950 BC in order to prevent mosquitoes. Herodotus, 450 BC, recounts the use of mosquito nets and the custom of constructing tall sleeping towers. Aristotle documented the long-standing use of fumigants by the Greeks in 350 BC, while the Roman Cato noted the use of oil sprays, oil and bitumen sticky bands, oil and ash, and sulfur bitumen ointments as pest control methods in 200 BC. Apart from all these techniques, Roman people would also recurrently turn to religion and superstition for help with their pest problems of plague, locust swarm, and plant diseases. One of several such mysticism, including a woman running on the garden and nailing crayfish in the different parts of the field, has been reported in the book on ancient Roman agriculture, De Re Rustica, 50 AD. The first use of the word pest also been mentioned in the ancient Roman civilizations, where traditional rites were performed by Roman people in the month of April to appease the goddess. 133. Robago. Robago was considered synonymous with the cereal rust disease and was given the name Maxima Segedum pestis, meaning worst pest of the period. 8.4 Factors affecting the abundance of insect pests. In his novel work, Charles Darwin, 1859, explained the idea of environment having various components which might act individually or in combination to influence the survival and multiplication of any living species. The basic components of food and other resources form the living or biotic factors, while climatic components made for the abiotic factors. In a similar way, physical and abiotic elements were discovered and used to explain how insects evolve and survive both as individuals and as a group. This was strongly confirmed by the effect of abiotic variables on the growth of insects, but the effect of several integrated components could not be fully understood for several decades. Regarding the density of the insect population, Howard and Fiske, 1911, provided an explanation of how these components function. They separated environmental factors into two groups based on whether their detrimental effects increased with population density, facultative factors, or were independent of density. Catastrophic factors. Nicholson, 1933, demonstrated environmental factors into two parts, reactive controlling, and non-reactive factors. These reactive factors were similar to facultative factors of Howard and Fiske, whereas the non-reactive factors were analogous to the catastrophic factors. Smith, 1935, described facultative factors as density-dependent factors and catastrophic factors as density-independent factors. Generally, the density-dependent factors are biotic in nature and the density-independent factors are abiotic or primarily climatic in nature. Any insect species that feeds on or harms crop plants can only be referred to as a pest if it causes significant crop damage or accumulates to a specific level of abundance in a certain location. Any insect species distribution and abundance are influenced by interactions with biotic and abiotic elements of the environment. Temperature, moisture, and soil nutrients, along with biotic interactions within and between species, can all have significant effects on how plants and animals are distributed in space. 134. Apart from the general biotic and abiotic factors influencing the reproductive success and dispersion of the insect pests, some major limiting factor can be categorized as the most important biotic and abiotic factors for the insect pest population. There are many factors that, alone or in combination, allow insect populations to reach densities that are of economic importance. Different such factors are following. Destruction of forests. Large-scale deforestation for the purpose of crop production creates favorable conditions for some insect populations to exponentially grow and turn into pests by altering the habitat's topography. Additionally, the deforestation-related changes in many abiotic parameters, such as temperature, diurnal fluctuations, humidity, rainfall, etc., have a significant impact on the insect population densities. Extensive monoculture. Any pest population of that crop plant species will have a suitable habitat if a single plant type is produced over a vast area. Since the pest has access to enough food and shelter throughout the year, intensive cultivation of such crops in more than one season in a year accelerates this population growth. Introduction of new crops and improved strains. Some natural insect populations that were previously not thought of as pest species may benefit from the cultivation of new plant types in a region where they are not native to the crop itself. Introduction of a new insect species in a new habitat. In order to control existing insect pest species as natural enemies, different bug species are introduced to new locations. The result is that the introduced bug species, which lacks any local natural enemies, proliferates in the area and begins to harm local economies. The best illustration of this type of outbreak was the introduction of the wally aphid, Ariosoma lanigerum, to the Nilgiri region, where it quickly established itself as a significant apple pest in the absence of any local natural enemies. Accidental invasion of a foreign pest. The pest outbreak is also made easier by the pest's entry into a non-native location where natural enemies are absent via one of the various transport routes. The most prominent instance of these invasions is the recent spread of the invasive alien fall armyworm in the Indian subcontinent. Host location and suitability. A series of steps is completed as an insect starts to settle into a crop, and the amount of time devoted for this purpose is influenced by the characteristics of the 135. Habitat of neighboring crops and species diversity of the area. The total time allocated to these steps and the number of insect species present in the same area determines the rate of colonization. These steps are host habitat selection, host location, host recognition, host acceptance, and host suitability. Reproductive potential. The rate of establishment and population growth of an insect pest into a crop plant species is substantially influenced by the life history characteristics of the insect pest population. For instance, a migratory insect pest species must have a high reproductive potential if it typically cannot withstand the harsh winters of a region. This results in the colonization of these species in high numbers for a brief period of time, as their life cycles ought to be brief as well. All of these life history characteristics are shared by both the locust swarm and the fall armyworm, Spodoptera frugiperda. Environmental limitations. In addition to the general abiotic factors such as temperature, moisture, air current and other calamities, pest population can also be checked by the natural enemies, residing in a new landscape or habitat. In case of biological control experiments, inundative release, the parasites or predators, released in the locality cause sufficient mortality in the pest population and reduce the colonizing population to a level that is far below the economic levels the following season. Furthermore, extreme fluctuation in the summer and winter temperatures can inflict high mortality on overwintering pest populations, effectively reducing the number of newly established insect pest population. Crop production practices. Different crop production practices like crop rotation, disturbed crop sowing and harvesting time and weeding can cause the pest population density to increase or decrease in any crop field. Pesticide application. Since ancient times, it has been customary to use chemical insecticides to clear afflicted areas of the pest insect populations. However, the use of such insecticides or other chemical pesticides can also present a chance for non-target insect pest species that are already present at densities far lower than those required for economic activity. When a pest becomes resistant to a wide range of insecticides and pesticides eliminate its natural enemies, the most severe outbreaks are likely to happen. 136. Another scenario of a sudden rise in insect pest density is brought on by the disruption of food chain, resulted due to the pesticide treatment. For instance, if aphids, spider mites, and thrips, the typical prey of numerous, omnivorous predators like Coccinella spp. and Chrysopa spp. are eradicated from the cotton agroecosystem by insecticides early in the season, predators will eventually starve to death, will leave their area, or stop reproducing. Later, when migratory pests like Spodoptera species and Helicoverpa species infest the areas that have been treated, they are essentially safe from predators, resulting in enormous epidemics of these pests. Host plant resistance. Cultivation of resistant plant species in new habitats or the switch from susceptible to resistant crop varieties in addition to the cultural practices have a detrimental impact on the abundance of insect pest populations. However, such cultivation also encourages the transition of a pest species that was once crop-specific to polyphagous or generalist pest species. Additionally, an existing problematic insect pest species may become even more diverse due to increased population density of a biotype in the population. 8.5 Types of insect pests. Insect pests have been categorized based on different characteristics of pest and its infestation. 1. Based on occurrence. I. Regular pest. Various insect pests favors certain crop and thus are closely associated with it, resulting in frequent infestation in similar crop and are called as regular pests. Example, thrips on chili and aphid infestation on mustard in North Indian conditions. E. Occasional pest. Such insect infestation occurs less frequently and are associated with more than a single crop. Example, mango stem borer, rice caseworm, etc. E. Seasonal pest. Such pests infest the host crop in certain season and month of the year are called seasonal pests. Example, hairy caterpillar, Amsacta albistriga, in groundnut and rice grasshopper, Hieroglyphus banyan. IV. Persistent pest. The insects which infest a crop almost throughout the year are called persistent pests. Example, mealy bugs on different crops, chili thrips, etc. V. Sporadic pest. The insects which are confined in few localities or isolated area are known as sporadic pests. Exemplaris earhead bug, Leptocoryza acuta. 2. Based on level of infestation. 137. I. Epidemic pest. Epidemic pests are those insects that suddenly appear in a severe form across a certain area at a specific period. Example: brown plant hopper, Nilipar botalugans, on rice in Tanjore. E. Endemic pest. Pest activity that is limited to a certain area and occurs on a regular basis in a few pockets at a low level. Exemplaris gallmige in Madurai, mango hoppers in Periakulam. 3. Based on damage boundary. I. Key pest. These are the worst and most destructive insect pests that can harm any crop. They have a GEP that is constantly above the EIL, therefore even the existence of the pest might result in financial loss. The occurrence of these pests is persistent. Example: diamond backmoth and cotton bollworm. E. Major pest. Such insect pests have a GEP that is near to the EIL, and the only way to stop the economic damage is by timely and frequent insecticide application. Example: rice stem borer, cotton jacid. E. Minor pest. Population density of such pests rarely crosses the EIL and GEP is always below. The economic damage. IV. Potential pest. Different insect population present at a crop field does not persist as major pests but can become serious pests with a change in abiotic factors. Example: Lepidoptera littora. 8.6 pests of stored grains. Several insect species are known to infest and be responsible for the huge economic losses to the stored products. The important species which are considered as the major pest of stored products are described following. 1. Rice weevil, Cetophilus oryzae L. It belongs to the family Curculionidae. The 4 mm long weevil has a dark brown color with 4 light reddish or yellowish spots on elytra. The female weevil bores a hole on the paddy grains, deposits a single egg and covers it with gelatinous fluid. The grub is opodous and feed and pupate inside the grain itself, followed by emergence through an irregular hole. The insect is a major and destructive pest of rice, wheat, corn and sorghum in field as well as in storage conditions. 138. 2. Capra beetle, Trogoderma granarium everts, family, Dermestidae, order. Coleoptera. The beetle is reddish-brown in color and measures up to 4 to 6 m long. It is a serious pest of stored wheat grains. Apart from wheat, the grub also feeds on oilseed and pulse seeds. Nature of damage. Larva feeds on the grains from inside, resulting into a hollow grain. This causes a rapid and serious loss of the produce. Larval exuvi also contaminate the product. Life history. Female lays 30 to 35 eggs in her entire lifespan of total 3 to 12 days. Larval development is completed in 18 days with male molting 4 times in comparison to the 5 times molting in female larva. During adverse conditions, larva can undergo a facultative diapause for 9 months. 3. Lesser grain borer or paddy borer, Rhizopertha dominica F. Family, Bostrichidae. Order, Coleoptera R. dominica is a serious pest of stored grains, particularly the wheat. The adults are more injurious than the larva. Whole grains may support several adult grain borers. It is thought that R. dominica is a secondary grain pest, however first instar larvae have been observed to enter grain through the intact kernel. The beetles have been observed to infest and attack other stored food commodities such as storage paddy, corn, millets, tobacco, nuts, beans, biscuits, cassava, cocoa beans, dried fruits, peanuts, spices and dried meats, fish. Larvae are voracious feeders. Identification. The adults are either black or reddish-brown and grow to a size of 3 mm. Their head is turned upside down into a hood, forming a triangle under the thorax. They have a distinctive shape, with a large pronotum. There is a prominent constriction between prothorax and mesothorax, giving the appearance of having only 2 body sections when viewed from above. Life cycle and nature of damage. April is the month of egg laying. As the adults live long, up to 500 eggs may be laid in total. The eggs are laid close to the embryo end of the grain, which is softer than other parts. It makes the young larva easy to enter the grain. Eggs are laid singly and in groups. Incubation period varies with temperature and ranges 5 to 6 days in summer and 7-11 days in autumn. The young larvae pass through 4 to 5 molts before pupation, which takes place inside grain. The adults emerge within a week and begin to feed on the grain. The minimum time to complete the life cycle under ideal conditions is 25 days, however generally it takes 2 months. 139. 4. Confused flower beetle, Tribolium confusum and red flower beetle, T. castaneum family, Tenebrionidae, order, Coleoptera. Both flower beetles are found throughout the world, attacking primarily milled grain products. The confused flower beetle predominates in temperate region, whereas the red flower beetle is more common in subtropics. Identification. Beetles are 4 mm in length and reddish-brown in color. The head and dorsal sides of the thorax are densely covered with minute punctures. The easiest distinguishing character is that the segments of antennae of the confused beetle increases in size gradually from the base to the tip, whereas in the red flower beetle the last few segments are abruptly much larger than the preceding ones. Nature of damage. Both larvae and adults damage the wheat grains and flour. The confused flower beetle is the most abundant and injurious pest of flour mills. The beetles attack flour and broken kernels, more severely during monsoon period. It also damages beans, peas, baking powder, ginger, dried fruits, insect collection, nuts, chocolate, etc. Life cycle. Each female deposits 400 to 500 eggs in flour or other foods. The eggs are white, translucent, sticky, slender and cylindrical and hatch in a week. The larva molts 6 to 7 times before reaching full-grown stage within 3 weeks. Full-grown larvae transform to naked pupa and a week later adult emergence takes place. The larva to adult developmental period completes within a month. Adults can live up to 1 to 2 years. 5. Pulse beetle or Chinese bruchid, Calosobruchus chinensis family, Bruchidae, order. Coleoptera, the pulse beetle, C. chinensis is native of China but is distributed in India, Myanmar, Japan, Africa, USA, Philippines, Malaysia, etc. Identification. The adult beetle is small and short, being 3 to 4 in length, stoutly built and dark brown in color. Head is small with rostrum and bears a pair of serrated antennae. The elytra are short and do not reach up to the tip of abdomen. Nature of damage. C. Chinensis is a major pest of pulses, particularly chickpea, pea, lentil, beans, etc. The grubs eat up the interior of the grain, leaving the outer shell of the grain. The beetle can thrive on milled whole pulse with husk, while it does not grow on processed pulse products. The beetles also infest the grains in field. Adults are short-lived and do not feed on stored products at all. 140. Life cycle. The female lays 40 to 120 eggs either singly or in cluster on the grains in stored conditions, whereas, in the field, eggs are laid on the pod and sometimes dropped loosely among the seeds. The eggs hatch within a week into white and curved larvae. The larva eating through the inner wall of the eggshell into the seed coat and then into the seed itself, where it feeds on the cotyledons. The larval period varies from 2 to 3 weeks. When fully grown, the larva measures 6 to 7 m in length and cuts a disc in the seed coat almost through and pupates below. The pupation period ends in 2 weeks. At adult emergence, the disc readily opens for adult to come out. There may be 6 generations in a year. 8.7 Pests of sugarcane. 1. Sugarcane leafhopper, Pyrilla purpusilla family, Lephopody, order, Hemiptera. The sugarcane leafhopper, Pyrilla purpusilla, is distributed throughout in India where sugarcane is cultivated. It usually severely damages the cane in Uttar Pradesh, Bihar, Punjab, Madhya Pradesh and Maharashtra. Identification. The straw-colored adult insect has 2 pairs of wings which are folded on the abdomen in shape of a roof. The length of the body is about 8 to 10 m. They possess long pointed snout with prominent red eyes. The female has a pair of pads on the abdominal end of the body. The adults are very active flyer. Nature of damage. The nymph and adults both damage the crop by sucking the sap from the foliage, depriving the plants with nutrients. As a result, the foliage of the canes become pale yellow and dry up. Like aphids, the Pyrilla also excretes honeydew, upon which sooty mold develops, turning the leaves black. Life cycle. The female lays eggs in large clusters, containing 10 to 65 eggs, which are covered with white waxy filaments secreted by the anal tuft of the females. Egg-laying site is the underside of leaves during summer and inside leaf sheaths during winter. The eggs hatch into nymphs after 7 days in summer and 22 days in winter. The freshly hatched nymphs are cream-colored, soon turning into pale brown, and have a pair of characteristic anal filaments. Nymphal period varies with climatic conditions. In summer it is about 6 to 8 weeks, but in winter it is about 17 to 18 weeks. The winter is passed in nymphal stage. 141. 2. Sugarcane shoot borer, Chilo infuscatalis snellen, family, Crampody, order. Lepidoptera. C. infuscatalis is a major pest of sugarcane in peninsular regions of India and more vital in early stages of crop growth, causing heavy economic losses. Additionally, the larvae also feed on oat, sorghum, millets, sugarcane, barley, corn and wild grasses. Identification. Adult moth is straw-colored with white hind wings and measures about 3 to 3.5 mm. The forewing is grayish-brown and have dark marking on it. Nature of damage. The pest attacks the crop in its early stages of growth with peak activity during March-June. Shoot borer larva enters the plant laterally in its third instar by making entry holes at the base of the stalk. The larva feeds on the soft tissues and makes cavities extending to the sets, killing the growing point. This causes a severe damage to the central leaf spindle, which dries up and form a dead heart that can be pulled out easily. The cut-off portion inside the bored plant decays, and the dead heart emits an offensive odor when pulled out. ETL for shoot borer is 15.0–22% dead hearts for late-sown sugarcane varieties, whereas, for early variety ETL is 16.8% dead hearts. Life cycle. The eggs are laid in overlapping rows, either in the basal or middle region of the lamina on underside of the leaves. After hatching in 4 to 6 days, the larva wanders for a few hours and reaches the base of the stem by crawling or hanging by silken thread. The larva enters the gap between the first leaf sheath and stem and feeds on the inner tissue of leaf sheath like a leaf miner for a few days. Later, the larva bores into the stalk and kills the growing point in 10 to 12 days. Larva passes through 5 instars. Grown-up larva is dirty white in color with dark brown head and 5 violet dorsal stripes. 3. Sugarcane root borer, Emilacera depressilla, Swinhoe family, Pyralidae, order. Lepidoptera. The root borer of sugarcane does not feed upon the root of the plant, but instead it feeds upon the oldest internodes present just above the mother or seed set. Identification. Moths have straw-colored forewings and whitish hindwings with 20 to 25 m in wingspan. The moth has dark lengthwise strip on each wing. The head of the adult moth is palipink while wings are pale or dirty brown. The lifespan of male moth is 3 to 10 days and that of female is 10 to 15 days. 142. Nature of damage. It attacks the underground stalk portion of cane, and the intensity of damage varies with the stage of the crop. In the early stages, i.e. during May-June, dead hearts are formed which cannot be easily pulled out, nor do they emit offensive smell as in the case of shoot borer. Life cycle. Flat, scale-like and creamy white eggs were laid singly on the underside of the leaves along the midrib. The first instar larva crawls to the base of the stem and enters it by making a single hole at or just below the ground level. The larva feeds in an irregular semicircular pattern and may leave the bored stem and enter into another underground stem of the same clump or adjoining shoots. Larval period varies from 23 to 43 days in summer and longer during rainy season and winter. The full-grown white-colored larva cuts an opening at the exterior end prior to pupation and constructs a silky pupal tube up to the surface of soil for easy emergence of the adult. Pupal period lasts for 7–11 days during summer and rainy seasons and 8 to 14 days during winter. 4. Sugarcane top borer, Sherpophaga excerptilis, Walker family, Pyralidae, order. Lepidoptera. Sherpophaga excerptilis is commonly known as top borer, sugarcane top borer, top shoot borer. Earlier, Sherpafog annabella was considered top borer of sugarcane. Identification. Adult moth is silvery white in color with or without one black spot on each of the forewings. The female has a tuft of crimson red, orange or buff-colored anal hairs. Nature of damage. Top borer occurs as a major and regular pest in the subtropical region but as a minor and occasional pest in the tropics. Capable of attacking the crop at all stages of growth, top borer characteristically produces leaf mines, shot holes on opened leaf, atrophied central shoot, stunting of cane and sprouting of axial buds leading to bunchy top formation. The unique early symptom of attack is tunneling by the first instar larva into the midrib, causing a white streak that subsequently turns reddish-brown and becomes visible on the ventral surface of the midrib. Life cycle. The white egg mass is laid on the ventral side of basal or middle portion of top second or third lamina in 2 to 3 overlapping layers, covered with the orange or dull golden yellow colored hair mass from anal tuft of the adult female. Newly hatched larvae crawl 143. Actively on leaves, suspend themselves by silken threads and get dispersed to adjacent plants by wind. After 3 to 4h, they bore into the midribs of leaves through the lower epidermis. The larvae prefer the most tender visible midrib, mostly that of the -1 leaf, which is rolled with only the lower epidermis exposed. The larva feeds above the growing point up to the 4th instar, after which it cuts across the growing point, causes dead heart and enters the top internodes, either tunneling down in the center or moving superficially towards the rind. Pupae are brown obtect and the pupal period varies from 6 to 8 days. 8.8 Pests of cotton. 1. Red cotton bug, Diastercis cingulatus, Fabricius, and D. Carnegiae, Fabricius, family. Pyrocoridae, order, Hemiptera. Both the species of red cotton bugs have always been a source of serious damage to the crop by feeding on developing cotton bolls. The red cotton bugs have been declared as one of the most destructive cotton pests in India and Pakistan and also in other parts of the world. Identification. The adults are deep red in color and measure 12 to 13 in length and have deep red legs and antennae. The membranous portion of the forewings and the eyes are black in color. There is also a black spot in each forewing. Nature of damage. They may start feeding with young flower buds, flowers and then young bolls. The growing bolls with buttery content in them are fed aggressively by a number of bugs. These fruits open prematurely, and they continue feeding on the seeds and immature lint, sucking the sap in them. In ripened cotton seeds, Bacterium Nomataspora gossipi enters at the site of injury and stains the cotton fiber. Due to this staining, they are also called as cotton stainer. Life cycle. During spring, the female lays the spherical yellow eggs in the soil and are covered with loose soil or with small dry leaves. Nymphs hatch out in about 7 days and after hatching are wingless. The nymphs feed gregariously and voraciously on the cotton bolls. Nymphs pass through 5 molts with wings developing from the 3rd instar and attaining full form after the 5th. Female nymphs are larger than male ones. 2. Spiny bollworm, Boreas insulana, Boystuval, and spotted bollworms, Boreas vitella. Fabricius family. Nullidy, order, Lepidoptera, spiny bollworms and spotted bollworms 144. Are the early season pests of cotton in India. They are seen from 35 days of crop growth in different parts of the country. Identification. The adults of E. vitella is small 10 to 15 in length and 20 to 25 across wingspan, having pale white forewings with broad greenish bands in the middle, whereas in E. insulana the forewings are completely greenish. Nature of damage. The initial infestation generally occurs on 6 weeks old crop, in which the caterpillar causes drooping and drying of shoot due to its feeding by boring into it. In the later stages the larvae feed on buds, flowers and bolls. As a result, flower buds and fruits drop prematurely. Fruits remaining on the plants get deformed and often show exit holes of the larva. One caterpillar is able to destroy several bolls until pupation. The infested bolls produce poor lint, having very low commercial value. Life cycle. The female moth lays 2 to 3 eggs on bracts, leaf axils and veins on the undersurface of the leaves at night. A single female may lays up to 300 to 400 eggs. The newly hatched larva bore into the growing shoots or bolls, consuming the plant tissue. The full-grown larva is about 13 to 18 in length and is brownish-white with a number of black and brown spots on the body, hence, called spotted bollworm. The last instar larvae come out the boll and pupate in tough felt-like silken cocoons, which is attached to dry leaves of the food plant or in soil or among the fallen leaves and rubbish material. The pupal period is about 10 to 15 days in summer, 20 to 25 days in autumn and 40 to 85 days in winter. In summer, total life cycle completes within a month. 3. Pink bollworm, Pectinophora gossypiella, Saunders family, Gulechiidae, order. Lepidoptera. This oligophagous pest is extensive cotton pest in all cotton-growing continents and countries. Possibly originated in the erstwhile Indian subcontinent, this Gulechiid insect has caused huge crop loss in both diploid and tetraploid cotton varieties. Identification. Adults have a 12 mm wingspan and are grayish or dark brown, with inconsistent black markings. The antennae are filiform and the hindwings are deeply fringed. The adult moths are nocturnal and fly around after dusk. Adults feed on nectaries under the cotton leaves and may live for up to 2 months. 145. Nature of damage. The damage to fruiting bodies of cotton crop is extensive due to the feeding of pink bollworms. Cotton plants produce 3 to 6 flushes of reproductive bodies in the active phase when the lint quality is at its best. The pest feeds on the seeds, affecting the growing lint. Larvae in older squares web the unopened petal rims and feed inside, causing rosetted flowers. Life cycle. Singly laid or in small groups of 3 to 5 eggs, flattened oval eggs measuring 0.5 mm long by 0.25 mm width. The eggs are laid in protected locations on the plant, such as axils of petioles, peduncles, lower side of leaves, old leaves at the junction of main vein, bracts, squares, flowers and 2-week-old bolls. 1 mm long, black-headed neonate caterpillars with translucent body hatch out. The neonates move to the nearest food substrates such as squares, open flowers or bolls and commence feeding on tissues. Second instar caterpillars have creamy white body, dark brown head and paler thoracic shields. Third instar larvae are 6 mm long and have creamy white body with 2 transverse dorsolateral pink streaks in each body segment. Those larvae that feed on flowers or on shed rotting fruit bodies have no pink color. Pupation is generally in soil. The mature larvae cut a round hole on bowl wall and fall out into soil. Pupae emerge between 8 and 13 days during cotton season. The grayish-brown adult moths with black bands on forewings and silver-gray hindwings emerge from pupae in early mornings or in the evenings. 4. Leaf roller, Coleopterygata fabricius family, Crampidae, order, Lepidoptera. Identification. Moth with yellow wings having brown wavy markings, whereas the larvae are bright greenish in color with a dark head. Nature of damage. Rolled leaves fastened with silken thread to make a trumpet-like shape is the most characteristic damage symptom of this pest. Large-scale damage gives a pale look of crop with rolled leaves and stunted growth. The pest frequently occurs in unclean farms with shaded areas. Life cycle. The moths lay eggs on the leaves, which hatch greenish-white, semi-translucent caterpillars. The larvae feed on the epidermis and cortex of leaves causing them to roll. Leaf cases formed by the feeding larvae act as the pupation site for an obtect pupa. The life cycle of the moth completes in 20 to 25 days. 146. 8.9 Pests of cereals. 1. Corn earworm, Helicoverpa z, Heliothis z, Bodie family, Noctuidae, order. Lepidoptera corn earworm is a key pest of baby corn in USA and is also found infesting the corn crop in different parts of India. Identification. Forewing yellowish tan with variable reddish-brown, olive-green, or gray markings and shading. Reniform spot usually filled with dark gray. Hindwing whitish with dark gray veins and border, with surrounds whitish patch at midpoint of outer margin. Larvae are translucent or yellowish-white in color with black head. Nature of damage. Corn earworm damage the whorl of sweet corn plants and the symptoms resembles that of early season European corn borer. Feeding is largely done on the developing milky grains at the top of the cob. In larger plants, caterpillar also grazes on the maize silk and eggs can also be found stuck in this silk. Corn earworm are cannibalistic in nature and thus are found singularly. Life cycle. Subspherical eggs are laid individually to the plant substrate, white to yellowish-green when laid, developing a reddish band and finally turning dark gray before hatching in 2 to 3 days. Developing larvae initially feed on the corn leaves, making circular holes, reaches to the cob where it feeds over the developing milky grains. Larva passes through 5 to 6 larval instars and pupates in earthen cells on the ground. Depending upon the season, pupal period varies from 10 to 25 days. 2. Paddy earhead bug, Leptocoryza acuta equals L. Varicornis family, Coridae, order. Hemiptera, it is major pest of rice paddy in different parts of India. The pest generally occurs during the flowering stage and feeds up to the milky stage of the crop. Identification. The adult is green, light brown or mixed yellow in color with a slender body and long legs. The male measures 13 to 14 and female 16 to 19 in length. Head is triangular and bears 4 segmented antennae. 147. Nature of damage. Adults and nymphs both suck the sap of developing rice grains at the milky stage and cause considerable yield loss. Sucking of the grain sap by this bug causes ill-filled, partial-filled and chaffy grains and also enhances subsequent fungal and bacterial infection. Life cycle. The females lays 250 to 300 eggs during night in 2 or 3 straight rows of 10 to 20 eggs along with the midrib on the upper surface of the leaf blade. Incubation period lasts for 6 to 7 days. First instar nymph is very small, nearly 2 mm long, pale green in color and look ant-like. It grows to deepen green through different instars. Wing pads appeared on the 3rd instar nymphs. Well-developed wings appeared after the final molt from the 5th instar nymph into adults. Total development period varies from 15 to 20 days. 3. Rice grasshopper, Hieroglyphus banyan fabricius, family, Acrididae, order. Orthoptera, it is a poppyphagous insect pest of several crops including pulses, millets, corn and rice. Identification. Dull green-colored hoppers with brown-black lower surface. Adult hoppers can vary from 18 to 40 in body size. Hindlegs are modified for jumping and head is hypognathous. Nature of damage. It is serious pest of rice in North Indian states. It is a sporadic pest but the level and intensity of infestation is severe in most of the incidences. Nymph feed gregariously on the germinated seedlings causing it to wither away. Adults on the other hand feed on the shoot and leaves but can also cut the earhead in severe infestation. Life cycle. Females deposit eggs inside the ootheca into the soil from October to December. Each egg pod ootheca contains 30 to 35 eggs in the wet sandy soil at 3 to 5 cm depth. The eggs remain in the soil till rains begin during the following June-July. The nymphs hatch out after the onset of the rainy season. Newly hatched young hoppers are brownish yellow and afterwards turn to dull green. After passing through 5 to 7 instars nymphs attain adulthood. Developmental period of females is more than males. Adults mate after 1 to 3 days after emergence and survived for 1 to 6 months. There is only one generation of the insect in a year. 4. Yellow stem borer, Sherpophaga incertulas walker, Triporiza incertulas, family. Pyralidae, order, Lepidoptera S. incertulas is distributed primarily in tropical Asia. It also 148. Occurs in temperate areas where the temperature remains above 10 degrees Celsius and annual rainfall is greater than 100 m. India, Nepal, Sri Lanka, Pakistan, Afghanistan, Nepal, Bangladesh, Myanmar, Vietnam, Thailand, Malaysia, Indonesia, the Philippines, China, Japan and Taiwan are the main habitat of this pest. Identification. The abdomen of the female is wide, and the tip is covered with tufts of yellowish hairs forming a circle, while the abdomen of the male is slender, and the anal end has a thin, hairy covering dorsally. Forewings of the female are pale yellow to dark yellow towards the tip or yellow with a pale orange tinge, and the discal spot is black and prominent. Nature of damage. Newly hatched larva enters into the leaf sheath, feeds voraciously for 2 to 3 days and then bores into the stamen, staying in the pith, feeds on the inner surface of the walls. When this occurs during the vegetative phase of the plant, the central leaf whorl does not